So hello everyone, I am Merve. I am actually a PhD here at Indiana University at Intelligent Systems Engineering, and I'm a member of FAMES Lab right now. Since I'm a PhD student in here, last semester I took Dr. Glazier's course and I worked with a CompuCell to make a model as a semester project. So I would like to improve that model and I will explain how I did my microvasculature CompuCell model. Yeah, thank you. So probably most of us are familiar with the vascularization mechanism. Actually, it is a combination of vasculogenesis and angiogenesis. Vasculogenesis is like the first attempt to create a vasculature formation. The main players are the endothelial cells and also vascular endothelial growth factors. This is like the very first time to make an existing vasculature. After that, the existing vasculatures try to make a sprout or try to make a vascular network, and we call it as the angiogenesis. Here in this schematics, actually we can see a lot of factors are playing a good role, not only the endothelial cells or the vascular endothelial growth factor, but also some parasite cells. They are like those yellow cells. Their role is to stabilize the existing networks. And also not only the VEGF as a growth factor, but also, for instance, the fibroblast growth factor is affecting the microvasculature system, stability, and the formation. So previously what I did is mainly on the endothelial cells and vasculature endothelial growth factors. So actually you can make a vasculature with only 2 of them. However, it won't be stabilized. So maybe after 24 hours, the existing vasculatures will be deformed. So actually the mechanism is much more complex than that. And in here you can see, and most of us probably know, that the endothelial cells have 2 types, as tip cells and stalk cells. And tip cells have a role to elongate and create a connection. So actually, I worked on the endothelial cell circle about being a stalk cell and a tip cell, because not always a tip cell remains as tip cell or stalk cells remains as stalk cells. They have a circulation. Sometimes endothelial cells can be tip cell or a stalk cell in the position. What I'm doing as a personal in my lab, I am working on the 3D bioprinting methods, which is making a tissue-like formation with using a bioinks and the cells. The bioinks can be a formula from the polymers, such as I can say collagen, gelatin, laminin, and so on, like the naturally occurring in the tissue itself. So those are extracted from the tissue, and you can make a different kind of chemical formulation to make a bioink to have a stable medium for the actual formation. So right now in my lab, I am also working on prepare some bioinks, which is suitable to make a microvascularization with the human umbilical vein endothelial cells. So I would like to model what I'm going to do as an experiment and see the parameter changes and optimizations and how I can find a suitable microvasculature in that kind of structure. And for me, like, I have to make a lot of experiments according to the bioink optimization, printing optimization, or microvascularization experiments. So CompuCell will help to see how it will be in a good position without doing too much experiments. So previously, I will have some functions as the chemotaxis, which is like a chemical movement through a high gradient of the growth factors to prepare a vascular formation. And also the diffusion inside of the matrix, the secretion of the growth factors. But the actual mechanism is much more complex. So actually those cells should grow, should have a mitosis, and should have a proper decay and the other behaviors. So I would like to improve according to that. So the mathematical background of it, that the CompuCell using an effective energy function, as Dr. Glaser explained before. This is basically on this cell contact energy, cell adhesion energy, and also those cells properties as the target length area and current length area. And also the concentration change according to the microvascularization formation. Each time it actually calculates the secretion, decay, and diffusion to find the local concentration. So what I did previously, like I first begin to see a chemotactic behavior, whether the endothelial cells will sense the high gradient of the growth factor and try to go there in a certain way to make a vasculature. But right now they are not making a tubular structure. They try to elongate, because to have a tubular structure, those elongated structures should have a flow inside and open that elongation to make a tubular inner hole. Right now they just try to elongate to where the high gradient of growth factor is. After that, since I am planning to use different growth factors as a different isoforms, they are actually preparing from the natural growth factors splicing, and the different isoform of VEGFs have different diffusion lengths. So as Dr. Glaser explained before, normally diffusion length is very short. I would like to have the longer as much as I can. So there are some commercially available isoforms. Right now I try to see which one is the dominant, like the chemotactic behavior, the high gradient. So one of them is like the red and blue. One is the longest one, the other one is the shortest one. So after that, I worked on the endothelial cell circulation, how they will be a tip cell or a stalk cell. So we can see the circulation with actually looking to the location of the cell. So if the location of the cell is like at the edge, so it has more contact surface with the matrix. So if you can find the common surface between the medium and cell in the CompuCell, so you can see that whether this endothelial cell is inside of the cell blob or it is at the edge of the vasculature. And if it's at the edge, then it can elongate and try to make a vasculature formation. So right now, since I am working with a 3D simulation, I don't have too much cell number in here. So in here, the blues are the stalk cells, which are at the inside, and the green cells at the outside. After a certain point, since I don't have too much number of cells, most of them are actually have a much contact surface with the medium. So they became to be green, they became to be tip cells, and they most of them try to elongate. Since I don't have any other cell type as the parasite cells or other growth factors, after a certain amount of time, the formed branches are actually collapsed, deformed. So I also would like to add the proper mechanism as the second supportive cell types and other supportive materials into the mechanism. And also I would like to work on more on cell cycle as mitosis of these endothelial cells decay and grow properly, and try to mimic the 3D printed structure that I am doing with a bioprinter to see, because with the help of this bioprinter, we can actually print the cells in a manner of a structure, or we can make a drop of cells which is located in a certain way. So you can actually see the behavior of the cells in that certain locations. And also, since I am also working on the bioink formulation from a different materials at collagen, gelatin, and so on, I would like to work on the links more to see the mechanical behavior change, because of the mechanical behavior of the bioink will change the cell's viability and the vascularization formation. So I would like to work on the links to see the mechanical property effects as well. Thank you. If you have any question or suggestion, I'm happy to answer. Thank you.
The number of cells you're looking at, and are you looking at, this is under the microscope, of course, right? So are you seeing this at single cell resolution, or are you seeing this at more millimeter scale?
We didn't do any mixture with bioink and cells right now. We are in that position. And like, I need to find the proper cell density of it, but you can imagine that I have a 10 millimeter to 10 millimeter printed structure, and I will mix the bioink with the cells. So in that 10 millimeter to 10 millimeter print structure, all those filaments will have a bunch of cells. So like not 100 or so, it should be more like in a cell density. But if we will do a drop print, then I can have a controllable count of the cells in a one drop. Then I can make a drop print with that and work on it.
So in your model right now, are you just using chemotaxis and elongation of cells, or you're also using the vascularization recipe that Roland Merckx developed with James? Only chemotaxis. Okay, so I think you can use both, I guess, and then probably, and in your references, I know that there is one work that's that you also should add there is by a previous student that we had here, Sotil Procopio, that he actually modeled the emergence of stalk cells and no, it's Sotiris Procopio. He actually modeled that in cells to be using delta notch mechanism to have cells, some cells to be, I don't know if it was notch positive or delta positive to be the actual cells that migrate, and the other ones to be the other kind of fate to be the ones that be the stalk. And then the cells usually kind of, as the delta pattern was evolving, some cells actually change place as being the cell that was steep, then became stalk, and a cell that was stalk become tip, and then it moves forward. He did that 2D, and it was a very simple one with just a bunch of cells here and a FGF source here that chemotax. But I think something that you should take a look as well.
So I think you again, I think for something like this, the key thing, you've got a few of the references here. The key thing is really to begin to assemble a list of all of the, there's been a huge amount of simulation of this kind of system. And so we really, the first thing is to try to pull together at least representative papers for the various mechanisms that have been hypothesized. Quite a few of them do use CompuCell, so those are repeatable. For example, Nicole Kleinsteuer's papers are repeatable, since she used CompuCell. And then from there, start by trying to replicate those results, and then move forward to the new geometry. A lot of those were in 2D, so going to 3D is a project. But I think as Julia said, there's been a fair amount of simulation of tip stalk cell selection. There definitely are, I mean, there's some biological uncertainty, scientific uncertainty about exactly how cells signal in vasculogenesis. And so there are different models that have different hypotheses about the mechanisms of signaling. But there are a number that definitely do generate good vascular networks. And so it would be important to try to work with people here. I mean, Giulio certainly knows those papers. And he could sit with you and help try to do that. But first, we need you to go back and try to dig out that sort of literature and try to frame those questions a little bit more clearly. I think the statement here is very good, but we would need to see what, essentially try to help you get replicate existing results to begin with. And actually, I don't know if he's coming today. TJ, who's new in the group, has done quite a bit of modeling of sprouting angiogenesis using the same methodologies, but not in CompuCell. And so I suspect he'd be pretty interested in trying to get his methods that were developed in a different package into CompuCell. And he has quite a bit of expertise in that. So I'm hoping he'll certainly be here some days this week. Whether he'll be here today, I don't know. Okay. Other comments or questions? Okay. Okay, who's next? I think we have 2 people left. So, Javier.
Hi, everybody. I'm Javier, and I come from UNAM. So that picture over there is the main library in UNAM, which is a pretty nice building to put it in the presentation. So I'm currently here doing a short research stay under the supervision of Professor Glaser. My background is actually glasses, and rigidity has to do with relaxation. So it has nothing to do with biology or CompuCell. So this is all quite new to me. So I'll try and, yeah. So I'll try and get the message out there to what I'm trying to work. So it's working with diabetic retinopathy, diagnosis and prognosis. So a little background on that is that, well, diabetes affects the ability for the body to use glucose, resulting in hyperglycemia. So this, you have diabetes type 1, type 2, and if you have a prolonged high blood glucose level, this damages your the blood vessels, and this may then produce blindness. Not just diabetic retinopathy. Just as time goes by, you get this. You start to lose vision, right? But there's great interest in finding a treatment for diabetic retinopathy. So what happens is that the vessels, in the retina, neovascularization appears as some vessels, they start to get no blood flow can pass through there, so you can get blindness from neovascularization. So that's so severe loss of vision, diabetes from neovascularization is related to peripheral retinal ischemia. So that's when you're not getting oxygen to certain areas of the retina. So the current treatment is, well, Professor Glaser mentioned it yesterday, involves laser, but this has also the issue that it makes you lose vision. So the patient may end up worse than before the treatment, because there's no actual treatment in which you know where you should focus the laser to actually fix the patient. So we could ask, is there a way we could make a treatment that would help people with diabetic retinopathy, well, actually don't go blind? So the issue here is that, well, you're dealing with a quite complex system, and you have a lot of variables which are they're actually interacting, and you have some feedback there. So you need to select your variables and try to produce a simple enough model, but it's actually quite complex, so you can get some interesting dynamics and, well, maybe model what's actually happening in reality, right? So the idea is that when you have Professor Glaser and one of his former PhD students were working on, is that when you have capillary occlusion, which this happens when you have diabetes. Leukocytes go into the capillary and don't allow the proper flux, so you can get capillary occlusion. Then you have hypoxia because the oxygen is not going to where it has to go. So then you have vascular endothelial growth factor increase. And when that, factor tells system to generate more endothelial cells. But when that increases, that can produce an edema, and again, an edema, capillary occlusion, and you have flux. You have like a certain loop there. So, of course, capillary occlusion can start from different things that I think this model is not taking into account because it will be rather complex. Of course, there's another important variable, which is angiogenesis, but that's also a very complicated thing to model. So we ignore that. And eventually, well, you cannot see it here, but it's neovascularization. So eventually, if this loop goes on and on and on, then eventually you have neovascularization. If you have neovascularization, then you can have blood vessels fill certain parts of the retina, which shouldn't be filled with blood vessels, and you can then go blind. So the idea, well, so that's more or less the problem regarding diabetic retinopathy. So the aim of this project is, like I said, to do a treatment for that, in which a patient goes in, they make some measurements, and then from those measurements, you can have a diagnosis and a prognosis. So where do we start? Well, you first start by making these measurements. The nice thing about this, well, not diabetic retinopathy, but working with the eye, is that you can see the vessels in vivo. So you have this, so you go and you can make this measurement with a fundus camera. Also found this on the internet, which is something you put on your iPhone, and you can measure, well, you can take pictures of the blood vessels in the eye. So imagine you have that. So now from those pictures, you need to take out all the information regarding, well, the fluxes, well, certain elements. So, okay, so before that, you need to, since you're going to work with the images, you need to skeletonize the images. You can use image J, and you have that. So you have here your blood vessels, basically. But from there, you need to infer a certain number of things like boundary condition, flux in terms of direction and magnitude. And you also, since this is curved topology, well, you need to infer if, I don't know, for example, if this thing is actually a loop, or in fact, this is one vessel and this is another vessel. So that's, so you get this, so from the start, you start getting these problems, issues that one has to be careful. So ideally, one would want to have the picture. From the picture you skeletonize, and from there you can have all that, you produce some sort of networks of pipes and nodes, and you also can compute it, which vessels are veins and which are arteries. And so you have the input, and once you have the input, then you can go ahead and put it on the 3D CompuCell. So that would be the workflow. You have the image. From the image, you infer the initial conditions, the boundary conditions, all that. You put it into CompuCell, and then from CompuCell, you get some output in which you can say in how much time does that patient will lose sight or so. So the good thing is that there's a code was developed by Xiao Fu and Professor Glaser. The thing is that this code is quite long. It has 3,000 lines, which is hard to it's hard to, I mean, it takes time to understand what the code does. But well, this is a workflow of the code, and what basically does, it generates, it generates the network, the nodes, the boundary condition. I mean, you input that to the code, so you have that in the code. From there, you compute the blood flow, and you go in this direction, simulate the pattern of oxygen tension. You simulate the pattern of VEGF, and then you enter this loop. So basically, you can ignore this part of this time divisible. So when you enter, you check. So since it's Monte Carlo steps, you in a number of Monte Carlo steps, you check if the VEGF factor is above certain threshold. So if it's not above that threshold, well, you just go in this direction and you start all over again. When that in fact happens, then you form a fluid portion near cap, which is basically the edema that forms a certain region, which is, it just has blood in there and it's not flowing. And you just keep going. You can go again and you start forming this patterns. So once when it reaches 28 days, you go out this loop, and then you do some sort of metropolis test in which you check for occlusion. So if you don't have occlusion, then you go back into the loop and you start and you keep on going. But the thing is that when you have occlusion, that means one of the pipes, you have to remove it. So if you remove one of the pipes, you need to start all over again. You need to, again, calculate the flux of oxygen, the VEGF pattern, the blood flow, etc. So that's why when that turns yes, you need to go back into this direction, and it's like starting all over again. So you can imagine that takes a lot of computational time. And well, the simulation right now stops when either there's no effective flow path, or you reach 3 years and 3 years inside the simulation. Right? So that's so right now I'm trying to get this project going, and I'm currently reading the code, understanding the code, trying to see if things could be optimized, and trying to have a critical vision onto what could be improved and whatnot. And well, that's so as you can see, this problem would be easier if you wouldn't have the blood flow. But the thing is that since you have blood flow, and from there you have the oxygen moving in certain direction. So if you have a region where you have occlusion, it doesn't mean that around that region, or if you remove a pipe from that region, it doesn't mean that around that region you'll start removing pipes, because you have some oxygen flow. So you can start removing from over here. So in that sense, you have non-local interactions. So that makes it quite complex and quite interesting. So current objective, understand the code. And well, that's all. And thank you. So that's part of the references, and this is the paper where the code is, the code and what they did is explained. So thank you. Any questions?
Mike.
So my question is, are these 2 or 3 dimensional simulations?
Well, it's quasi 2D because it's on a plane, but you have certain space in the C direction.
Any other comments, questions? So this is sort of a different example because we're starting with an existing simulation, rather complicated one, and we're trying to now back out how to make it work. as Javier pointed out, there are a couple of big issues. One of which is that the simulation is written isn't designed to take experimental images and import them, which you'd have to do if you're really going to use it. And the other one is that to start with, the simulation assumed a limited number of biological mechanisms, which may or may not be the ones that are most relevant. Yeah. Yes.
Actually take a picture of a patient,
Mhm.
use CompuCell, and then try to make some sort of analysis prediction as to how to proceed with treatment.
Yes, the goal of this is that we actually will use this to improve therapy, because current therapy is not terribly optimized, and the therapeutic intervention is computer controlled. And so if you can tell the laser ablate here, that's actually a relatively trivial modification of the existing machines. It's not a trivial modification from the point of view of the FDA, but it's a trivial modification technically. And so if you had a computational method that said, instead of ablating in a grid or a random pattern, that you should ablate in particular regions, that could be a software patch to an existing machine.
So right now they're just doing what you said, a random pattern or a grid?
Yes, that's right. There's no calculation of maximal risk. And the problem with that is, think about how people fight forest fires. You build fire breaks. And in cities, you do the same thing. You demolish houses to block fires. And that works. But imagine now that instead of doing your fire break where you think the fire is going to spread, you just randomly cut down trees, or you just randomly demolish blocks. Well, in that case, you're going to demolish a lot of things you didn't need to demolish. And there's also a substantial chance that you didn't actually block it because you didn't block the things that were important. And so at the moment, there is no calculation of where you should actually do the ablation.
But I imagine one of the parameters is strong ablation or weak ablation, right? Or that's
It's pretty much binary at this point. The size of the ablation region is something that could be controlled. And so at the moment, they use fixed size ablation domains, but you could in principle have finer or bigger ablations. At the moment, they're more or less round or square. You could in principle draw lines to fire break. And so, I mean, to me, this is actually, if we get this working again, this is a place where you could, in 3 to 5 years, you could actually see a technology that was in the clinic. We've actually talked to the FDA about if we ever get it running, what it would take to get it licensed. We've talked to the big clinical trial people about getting the data, which they're happy to give us. But again, all depends on having somebody doing it. So, as I said earlier, Xiao is a great student, but he graduated and he's doing something different now. And so, but if you wanted an example of something that really could have clinical payoff, I think this is probably the one that's closest. I mean, I think a year worth of full-time work on this, you could have a clinical product. That's not in the clinic, but you could have the software ready for clinical testing. Yeah. But it's not easy to get back into it because it's a big package. The biggest thing that is missing in CompuCell from the point of view here, and it actually came up a little bit yesterday in the discussion of the lung branching, is that CompuCell doesn't have built in a fluid solver. Now, there are different kinds of fluid solver. You could imagine doing a full Navier-Stokes solver, coupling something like Phoenix, which does fancy Navier-Stokes. And Roland is coupled to some solids modeling packages, not our code, but his code. And so that's possible. We've talked over the years about, say, trying to couple to FE-Bio, so you have better mechanics. In this case, all the fluid solving is done using a simple Kirchhoff-type solver, which is relatively compact. But it's still writing a Kirchhoff solver is still hundreds of lines of code, just a Python, standard Python. And so that's the kind of thing where if there were a lot of people using it, the goal would be to turn that into C++ code rapid and then have simple calls so that instead of needing 500 lines of code, you'd have one call which says, this is the vascular structure, these are the rules, and now calculate it for me, to hide that from the user. At the moment, that's all done out by hand. Another example of that, which you're not going to see, but which was just mentioned briefly, was in the, I showed some simulations earlier of segmentation. Actually, simulations that Giulio and Susan wrote a long, long time ago, before the SPML CompuCell integration was done. And so in that case, all the molecular network stuff was solved by hand in the code. And you got the same answer, but the code specification then was thousands of lines or many hundreds of lines because you had the solvers written out. And so the goal is, so that was one of the big motivations for putting the SPML integration in, so that you had a compact way of specifying molecular networks. At the moment, we don't have a compact way of specifying flow and transport, something we're thinking about. Andy has some ideas about this in his own code. Whether that'll ever wind up inside of CompuCell is another question. It would be nice if it did. But the key thing there really is to try to use it enough by hand that you understand what it is you want to abstract. Because when you abstract these things, you reduce the flexibility of the solvers, because you have the things that are easy are the things that you've decided you're going to make available. And so you want to think very carefully about what people want to do and make those the things that are available. People still can do anything. Of course, they can always write their own Python. But you'd like to take those things and make them easier. And so this could be a good example. I mean, if you're interested in this project, we'd love to have people work on it. So.
Is there an experimental component to this?
Right at the moment, the answer is no. But as I say, we've talked with the main clinical trial people at Emory, and they're, if and when we're ready, they're happy to share clinical data on it. The eye is a funny thing because it's one of the few areas where it's easier to work in humans than animals. Because the, I mean, I don't know about you. I don't usually like going to the doctor. I love going for eye exams. Basically, as an engineer, I love the machinery they have. The equipment is wonderful. But it's extremely optimized to the human eye. And so if you're a mouse, the geometry of your eye is different. And there aren't the imaging technologies that are available for mice or rabbits, or you can do it on pigs because they have eyes that are, you know, humans. But it's also, you have to tell them to not move and this kind of thing. Right, exactly. So it winds up actually in the retina, you have lots of human data. And a lot of it's not invasive, but of course there are things you can't do. Here at IU, our optometry department has some very, very good technologists. And one of the things that they've developed, which we'd love to get back to access to, is using adaptive optics imaging. So they can actually image flow, not just structure, but flow, because there's this ambiguity in the structure images. And they can actually do it in 3D. So they can go deep into the retina. They can even go behind the pigmented retinal epithelium and image the vasculature behind the pigmented retinal epithelium. They need to use a tracer, so it's not totally non-invasive. They have to use a fluorescent tracer. But that's doable. And again, if you have but the trick, of course, there is you need the IRB to get, you have to have this ready for that. But in principle, you can go in human subjects and actually image the flow in surprisingly good detail in 3D and use that to validate this kind of model. The one thing that's hard these days is the following, which is people with diabetic retinopathy are always treated, or almost always treated. And so you don't have a good control group for watching what happens to untreated patients. The data that's available for that tends to be pretty old. Right. Right. Yes, they do. Right. Oh, yeah, the maps are there. Oh, yes, there's a lot of data. One of the things that's so discouraging if you read the clinical trials, while he's setting up, I can talk, is that if this worked, you would treat once, and it would work. And instead, the clinical trial protocol is, well, when the patient's coming in for their 6th ablation treatment, to do this, which indicates that fundamentally the ablation is not blocking progression. Because if it did, you wouldn't have to go back. And so I think there's a perception on the part of the clinical community they really need to do better. There is an alternative therapy, which is using Avastin, so a VEGF blocker, which is also used for the age-related macular degeneration. And the problem with Avastin is it blocks all blood vessels growth. So it damages everything. It also, of course, is pulsatile because you can't give it continually. And so it's not a wonderful treatment either. But the current best treatment protocol is actually a combination of ablation and Avastin. No. Unfortunately, it's not very effective. It's not, I mean, it causes a lot of damage and it slows things down, but it's not by any means a cure. It's a little bit like some chemo cancer therapies. I think we could do a lot better if we understood the mechanisms of the disease. And so this is, I think, actually a place where this kind of thing really could have real payoff. And I guess legally, I have to say, we've got patents filed on all this stuff. So there is some, as a conflict of interest disclosure, we do have IP on it. So we should probably put that in our slide deck. In my slides, it's there. He's new, so.
All right, so hey guys, my name is Jamie Adams. I'm a first-year PhD student from the University of Sheffield in Kira Campbell's lab. And for my PhD project, I'm basically looking at how a specific set of cells within the Drosophila embryonic midgut undergo an EMT or an epithelial to mesenchymal transition, and then undergo a collective migration until they eventually form the midgut. Cool. So just to give you an idea of what I'm looking at. Okay, sweet. So yeah, this is a Drosophila embryo. So this is a bit later than I usually look at. I like to show this because it's quite pretty. So it's quite easy to understand when you work in Drosophila. But, perfect. And then the 4-lobe structure you can see highlighted in red ends up being the final gut in the Drosophila embryo. So it's actually quite large in comparison to the rest of the embryo. So it's very easy to work with as well. So what do we actually see? So as I said, I'm looking at how cells can undergo an EMT and then undergo a collective migratory event. So I'm interested primarily in the posterior midgut primordia that form the final Drosophila embryo. And this is simply down to the fact that the posterior midgut primordia only require one master inducer to undergo EMT, whereas the anterior portion, which is down here, require 2 separate EMT inducers. So in terms of like genetic manipulability, it's actually it's far easier to work with the posterior. So yeah, so initially they undergo an EMT and then they'll undergo a collective migration, which is a huge event given the size of the embryo. And then they eventually meet up with the anterior portion of cells and they'll re-epithelialize or undergo an MET, a mesenchymal to epithelial transition, and form a solid embryonic midgut. So just to give you an idea of these cells I'm interested in, where they're actually sat within the embryo, before they undergo differentiation and active migration, they're just sat in this small little pocket just at the end of the germ band. So yeah, just to reiterate, my main interest is the period in which they undergo EMT and then migrate. The inverse of this, the mesenchymal to epithelial transition, I'm not that bothered about because that's a different project within the lab. And again, just to reiterate, it's only the posterior primordia that I'm interested in, so the cells highlighted in green here and not the red. So just to give you guys an idea of what I actually work with, I've got some nice videos here. So this portion at the back here, the posterior midgut, you can see migrates round, and eventually meets the anterior portion. And then you can see where they've met in the center here, where there's a difference, almost like a seal between the two, and then they re-epithelialize and form the midgut. So another interesting thing about the posterior midgut primordium is the fact that it's a heterogeneous group of cells, and there's three main cell types. So you've got the PMEX, which are these more columnar cells at the top, which are the primary midgut epithelial cells. You've got the ICPs, which are highlighted in red, and they're the interstitial cell populations. And then you've got the AMPs on the far left, highlighted in green, which are the adult midgut precursors. And so once again, to give you an idea of what this actually looks like in real life. So you've got your PMEX on the borders, and you've got your ICPs in the center, and then your AMPs lagging towards the rear. So I've got another video in a minute, which will show that a bit clearer. Because throughout our research, we've also found that there's a layer of mesoderm surrounding the endodermal PMEX and ICPs and whatnot. And this mesoderm is actually necessary for the PMEX to undergo migration as well. So I've got quite a nice video to highlight this. So before I play it, I'll just show you. You've got the mesoderm in green at the top and the bottom there. And then you've got the PMEX, which are the more red columnar cells. And then you've got the ICP cells towards the center, which are larger. And then you'll see the AMPs kind of sitting at the back when I play the video. Yes, yeah, exactly. But there is also, so there's an active migration for a period of about half an hour, but then there's also germ band retraction, which actually pulls everything in the embryo around much faster, which is problematic, which I'll talk about later on. So we know that cell plasticity underlies actual midgut morphogenesis, and this is because we've done a number of experiments to prove this, but we don't actually know why the cells migrate. We know that they'll undergo different morphological changes. They change their polarity, they gain mesenchymal characteristics, but still maintain some epithelial characteristics, but we don't know why they're actually migrating. And this is why I want to use the modeling software. Yeah, it's a bit misleading, no. So I've got something later on which shows this more clearly, but essentially all we have to focus on for now is just the highlighted areas. So these are cells initially, epithelial cells, but this is actually misleading because this looks like this is supposed to represent kind of the outside, the ectoderm, but the endoderm is not derived from the ectoderm. But so there's an image later on that I'll show you that'll make that, it'll be much clearer. No, it's not a tube, but you've got layers of cells and then they're kind of blocked in physically by amnioserosa, which the cells kind of move under and push out the way, and then an ectodermal layer on top, which acts as another like physical restraint. Yeah. So how do we know what I've already spoke about so far? So we know the serpent, which is homologous to human GATA4-6, drives the EMT, and that's solely responsible for EMT in the posterior midgut primordia. We've shown this through a number of experiments so far. We've generated serpent mutants, so serpent knockout embryos, and we found that crumbs, which is an apical-basal polarity regulator, which we see is knocked down all the time whenever cells undergo EMT in the posterior midgut. We've shown that's still expressed in a serpent mutant, and we've also shown that E-cadherin, which is a molecule necessary for adherent junctions between cells, is also highly expressed in this epithelial group. And then we've also looked at a number of different polarity markers, such as stranded at second, crumbs, bazooka, and F-actin and DLG, and also E-cadherin to determine whether things are downregulated, and looking at whether apical-basal polarity is maintained, whether adherence junctions are maintained as well. And so here is a panel showing that basically before EMT occurs, you have high and localized expression of crumbs, stranded at second, bazooka, and DLG. During, it begins to be downregulated, and then afterwards you have almost no expression of crumbs, stranded at second, bazooka expression, and DLG are both, they both become delocalized as well. Interestingly though, we found that ECAD transcription is downregulated. However, we do find dynamic punctate of ECAD in cells that are migrating. So we think that ECAD is expressed, which helps to maintain transient adherence junctions between motile cells during migratory events. So here's another little video to show. So once again, this is the posterior midgut primordia, and it's starting to undergo EMT. And what you're seeing here is as the light fades, that's showing E-cadherin, is showing that as cells start to move and transition into a mesenchymal state, the puncta become more dynamic. The ECAD puncta, sorry, become more dynamic. So from this, we developed a basic model so far in which we show that serpent is necessary to force cells to undergo an EMT. This directly inhibits crumbs, and then we also have upregulation of a number of different factors such as matrix metalloprotease 1 and forkhead. And this follows a range of different characteristics such as certain cells losing apical basal polarity, fragmented or transient adherens junctions, and we also have massively increased motility and invasive capabilities. So another thing that I think is interesting is that the ICP cells in the center here that form kind of the meat of a sandwich essentially, they have a large number of protrusions. They've got loads of cytoskeletal like extensions, and most of them, if not all, come into contact with other ICPs at certain points. So these cells seem to be actively extending protrusions to touch one another as they're migrating. So this hopefully should clear up that earlier diagram a little bit. So once again, just to show, we've got a system in which we've got mesoderm outside the gut, and then we've got the endodermal tissue. But so if you take this image and kind of rotate it so you're looking at it from behind, you've got the ectoderm layer on top, you've got the amnioserosa on the bottom, and then you've got the mesoderm surrounding the rest of the gut, kind of making a tube, and then you've got the endoderm that migrates through that tube. And so in our system, we've seen that our ICPs undergo proliferation quite regularly. However, there's no apoptosis. Our cells are extremely organized. They always form this same layer in terms of mesoderm followed by PMEX and then ICPs. We've shown that we know there are physical constraints in terms of the ectoderm and the amnioserosa. And we know that there's a mass conversation between different cell types. So we know that the PMEX and the ICPs have a conversation with each other through ECAD, to transient ECAD adherence junctions. And we know that the PMEX and the mesoderm talk to each other through integrin connections such as PS1 and PS3 and PS2. However, this isn't the inverse. If you ablate genetically the mesoderm, the PMEX won't move, but if you ablate the PMEX, the mesoderm will still migrate happily. And if you ablate the ICPs, the PMEX will still migrate happily, but if you remove the PMEX, the ICPs won't move. So this should hopefully also, I think I described how the cells actually look quite poorly, so this might help. So you've got the mesoderm in green, and some of the blue is specific mesoderm expressing FAST2 as well. Then you've got the endoderm in red. And so hopefully this video should show. So as we go through the embryo, you've got the endoderm in red there, and you've got the mesoderm surrounded that in blue, amnioserosa in the center. And as you come up through the embryo, you get to a point where this blue will overlap a little bit, and that shows that the mesoderm is kind of almost overreaching, leaving you with just a layer of amnioserosa and then ectoderm on top. So what can we actually measure from the system? So looking for us, it looks like the ICPs and the PMEX have relatively large nuclei. So in terms of tracking, it's relatively easy. And for now, we're not very interested in the AMPs because we don't think they do much currently. So this is just a stereotypical video of what we'd have to track. So I've only shown you a small portion of what we actually do track because of the issue in which you have the germ band retracting background, because after this, the whole system speeds up, but we're trying to think of ways to work through that at the moment. Yeah, so we've also tracking, we've got a custom macro that allows us to track in 4D, which is quite nice because we can track cells that move up through Z planes as well as across an XY axis. And from this, we can just get some very basic parameters such as your velocity, your coordination, which we measure as the angle between one cell and its closest neighbor. So 1 is moving perfectly perpendicular. 0 would be if you're moving at a right angle, and -1 would be if you're moving in the opposite direction. And then also just directionality or persistence, which is from point A to point B in the most linear fashion possible. So in terms of actually making this into a model, none of us have ever done any modeling in the lab. I've only just learned how to code this year, so we're all very new to the system. However, from what we have so far, I think that we have enough to fulfill the equations for the velocity and adhesion in terms of the cellular POTS model. And I've also recently made a cell counting macro in R, a script that I think I might be able to help with the volume. However, as I said, we've never done modeling before, so this is actually something that I'd like help with while I'm here, learning which parameters to input into the system and then how I could measure them, or what's the best way to separate these parameters into different categories. So my current plan would be to build a very basic model, set off either the mesoderm and the PMEX and the ICPs, and basically see if these cells can be modeled to migrate as we see them migrate in an actual biological system. And then if we can do this, see whether certain assumptions are true that we currently have in terms of how the cells are moving and why they're moving. And then as I get better at this, or we measure more different parameters, develop a more in-depth model. That's it. So thank you for listening. If there's any questions, I'll happily take them. So our assumptions at the moment are that in terms of the cells migrating through the tube, there's no pushing force from behind, because if we genetically ablate the mesoderm, the cells don't migrate at all, but we still have germ band retraction and extension. So the whole embryo still carries on as it normally would, but it's just the posterior midgut primordia that stay in that small niche that I showed at the beginning. And we don't know if there's any chemotaxic effects, but so far from any of the microarrays we've done through the gut cells and in the mesoderm, we can't find anything that we think would be massively upregulated and would be chemotaxic. So at the moment, we have no idea why the cells are migrating, which is why I want to try and model it.
So you say you don't think they're being pushed, but they could be mechanically being pulled.
Yes, yeah.
I mean, because one of the problems is when you see something like this move from here to here, you don't know if it's something that's being pushed from the outside or if there's something internal that's actively migrating it.
Yeah.
I mean, if you look at a train and a subway car, right, the train, you have a locomotive at the front and the cars are passive. In a subway, every car has its own motors, but the movement of the train looks the same.
Yes.
And so that's a tricky one. When you see this kind of bulk movement without a lot of reorganization, one's instinct is to say that this is because of some outside mechanical forces that are pushing on it. Now, you're seeing sliding relative to those mesodermal cells on the boundary.
Yeah.
And so that does suggest you have active migration. And that's something you can think about. One thing that would be very valuable is how much, if any, ECM is there in this tissue? Because when cells move, they have to pull on something, and they either have to pull on ECM or they have to pull on each other.
So that's a project we actually have ongoing at the moment, because in Drosophila, it's thought that the hemocytes are the things that deposit the ECM constituents, but that's not until later on, until about stage 14. So this is all from stages 9 through to about 11. So we think that there's an ongoing project that we think the endoderm might actually be laying its own tracks,
right,
in terms of like a train moving is laying its own tracks, and then going across the tracks, it's laid itself in terms of just laying laminins.
So I would say the place to start in terms of a pattern for the kind of modeling you might want to do, again, would be, I mentioned it yesterday, is this modeling of Kulesa for collective migration of neural crest cells.
Okay.
It won't be identical, but if you could replicate that in CompuCell, then you'd have something that might be.
Yeah, I just want to learn the basics at the moment, to be honest. Just learn how to use the program.
And there are a bunch of, questions. This system is very large and very complicated. You could ask the question, for example, how are the cells differentiating or de-differentiating? How do you build the architecture of that tissue with the layered structure?
Yes, yeah.
And then a third question is, once you've got that architecture, how do they move? And so one thing to do is to try to break this problem down into simpler problems.
Yeah.
Because there are a lot of issues. In other words, the movement, you could say, well, we're just going to have two cell types, which is a boundary cell type and some cell type in the middle.
Yeah.
And we want to get the middle cells to move relative to the boundary.
So that would be my first instinct, to put the PMEX and the ICPs together and then just try and model their movement together.
Right. And so then you could perhaps, for example, Giulio has this idea of filipodial links and pulling. And I think Priyam has done, she's not here either, unfortunately, but she's done some work on cells crawling on epithelial surfaces. Okay. But I think you may have to go relatively far from this specific problem to think about how to simulate the specific mechanisms you're seeing.
Yeah.
And try to understand those one at a time before you try to put it together. But I think Julio's thought a lot about some of these migration aspects. And the key thing there is how do you move relative to a surface? And that happens to be, we saw some vertex model simulations yesterday, where I think Stefan showed them. Vertex models tend to be particularly good at doing that particular kind of modeling. In CompuCell, you can do it, but you have to work a little bit harder to get that to work. But again, we should sit down and go over some of the details here. There's a great outline. And you may, I mean, initially the mechanical model may not have much molecular information in it. And then you may have a separate molecular model which talks about differentiation of EMT from a molecular point of view, the regulation of that. And so, but I think it's been very interesting. It's certainly be nice to see some Drosophila simulations. I mean, a lot of the things you're looking at here are not that different in terms of what's happening morphologically from what happens in segmentation. We didn't, in our segmentation, we didn't really do axis elongation. We sort of skipped that. But in principle, you also have this kind of axis elongation and reorganization, say during notochord formation.
Yeah.
And so there's some shared interest there too. Okay. But of course, we heard yesterday somebody else who was interested in EMT issues.
Yeah.
So again, there's a fair amount of overlap of interests here.
I've been coming to this workshop for a couple of years. I'm a little bit different than a lot of people here in that my background was pure biology, wet lab, experimental biology, and I became interested in modeling. Now I'm a postdoc in the future of work and learning, which means basically I'm supposed to be in two places most of the time. And so that's why I'm running in and out. But anyway, I want to kind of I'm going to try to finish a model I started in graduate school this week. And I also, at the same time, want to talk about some of the issues that kind of wet lab biologists face when they try to interface with modeling approaches, because that's one of my research interests. So my dissertation lab was a molecule of interest lab, like the vast majority of molecular biology labs are. We studied the alpha-2 beta-1 integrin. My PI had been funded for 30 plus years studying the alpha-2 beta-1 integrin and nothing else. And that remains the case for a lot of labs. And so, you know, interfacing with that is important as we talk about developing modeling tools. Part of my dissertation research was on trying to understand this phenotype. On the, can you see the mouse? Yeah, so this is the wild type retinal vasculature. And on the right, you have the retinal vasculature of the knockout mouse. And so you have greater lacuna between the vessels, and you have a high degree of vascular irregularity in terms of the level of angiogenesis. So it's a relatively subtle phenotype. And this has some similarities to several retinal vasculatures that have been phenotyped in Notch mutant animals or Notch mutant mice specifically. But it doesn't have the exact same overlaps. And so a lot of the time what happens in molecular biology labs, as you know, is you see a phenotype and then you find a recent high impact paper and you try to kind of integrate it within that paradigm. And that's what we did. And around this time, there was a very interesting paper, and I think this is maybe one of the first kind of high impact papers that argued about tip cell interchange, or kind of demonstrated tip cell interchange using a number of complex experimental systems. And they had modeling in there too, but the model was not available for reproduction. It was an agent-based model, and I'll talk a little bit more about that later. And at that time, recent experimental data from the zebrafish system had suggested that our molecule of interest, alpha-2 beta-1 integrin, was an upregulator of delta-like 4, which is part of this delta notch signaling system that had just been implicated in in tip-notch signaling. And so we conferred with a mathematician about how we could model that. We were redirected away from agent-based models to kind of a continuous model system in MATLAB. And so we adapted some work from Collier, and I mean, it's been several years now, so I don't remember all of the authors that we borrowed from, but we had a PDE model for the calculation of the amount of VEGF, and then based on that, tip cell and stock cell percentages were there. And we were able to kind of simulate a 2D retina. I don't really believe these simulations. I think that the model is pretty overfit, but under certain circumstances, you could kind of see how the phenotypes appeared similar to what was seen or what we observe experimentally, even though it's probably for very different reasons. This is a very underpowered model. And coming from a biology lab and working with pure mathematicians, we didn't have a really good idea of how to kind of systematically evaluate some of the phenotypes we saw. But we published it anyway in a, in a relatively minor journal. And now I'm interested in kind of taking this. So the story here is that the retinal vasculature predicted from the model based on our understanding of the role of alpha-2 beta-1 integrin in delta notch signaling was almost diametrically opposed to what we actually observed. And so we did a number of experiments, which I'm not going to bore you with, that basically showed that yes, alpha-2 beta-1 does support DLL4 as other people had previously reported, but that it also may interact with the delta notch signaling by repressing VEGFR1, which is a competitive, competes with VEGFR2. So in this way, it's possible that alpha-2 beta-1 integrin has both a positive and negative effect on delta notch signaling. And so it's possible that the integrin could act as kind of a rheostat. In high growth factor signaling environments, it could have one effect. In low growth factor signaling environments, it could have another effect, depending on whether it's promotion of DLL4 or its inhibition of VEGFR1 was the dominant factor. So the local research question is to kind of understand is if to try to build a model in CompuCell that's more biophysically realistic, that tries to determine how or whether a molecule acting in this way at 2 points in the delta notch signaling system can have this kind of rheostat function as this differential a phenotype in high and low growth factor environments. And then this kind of leads to a meta research question of, can we build an angiogenesis interactive that people can test their own signaling hypotheses in? And then now, a lot of the people in the biophysical modeling system don't actually believe the delta notch hypothesis, and I'll show you a little bit more about that. And so this leads to kind of the other research question, which is that by building this type of interactive, can we get experimental biologists that are highly focused on cellular signaling to think about how their signaling networks interrelate with and integrate with biophysical mechanisms. And so in terms of what modeling efforts are there, this is, James has mentioned this a couple of times, but Roland Merks published a model that shows, or that he argues shows that tip cell overtaking, that process where tip and stock cells switch, or where a new tip cell overcomes the previous tip cell. Roland Merks argues that that can happen purely through contact inhibited chenotaxis. And then at the same time, there's been continuing publications by that group I showed previously, but Katie Bentley is the primary modeler in there, further refining their argument about how kind of molecular regulation of delta notch signaling and tip stock signaling affects angiogenic phenotypes. And so these bodies of research aren't really talking to each other the way they should. I've been wanting to, kind of finish that aspect of my old work for a while. I've tried to replicate the Merck's model. It works. I can't get the stepables to work in it. you know, that is probably my fault. I've tried to replicate the Bentley model, but they have been kind of studiously, you know, kind of obscuring kind of the implementation details of their model, including having typos in many of the ODE equations for their papers. That's where I'm at. Yeah, they become right. So I think that's Roland's argument, that the cells changed position because of this contact mediated chemotaxis, and then whichever cell happens to be in the tip position takes on this tip cell phenotype because of greater exposure to endogenosis. So that is, of course, very difficult to prove experimentally or modeling wise, whether that's actually what is happening or not. Yeah, DLL4 is what's in the tip cell, and then there's more notch NICD expressed in the stock cell. It's all
Okay, so the tip is delta positive and notch negative.
Yes.
Okay.
Yeah.
Right. Yeah, so maybe you can try that in the exercise. Make all the cells in a line and see. Show that, you know, if it's the inverse, then we have a problem, of course.
Yeah, I think when you do the delta notch exercise, in the past I've just replicated, but I'll change the geometry and try to you know, try to do it that way. I think that'll be interesting.
So you're not able to replicate any of the angiogenesis, even the simplest ones?
I am able to do the ones without signaling.
Okay.
I was unclear about that.
Okay.